Let us start recording and welcome to the course BTL 602 Cell and Molecular Biology. We shall discuss today on plasma membrane and its structure. Plasma membrane is an important and essential component of cell. That means cell has a well-structured membranous envelope. So cell membrane structure and functions are very critical for understanding the cell biology or molecular biology. So plasma membrane, it is the boundary that separates the living cell from its non-living surroundings. Some cells are, unicellular, and some are multicellular. Whatever it is, cell membrane is a boundary of a cell, living cell, and it involves in communication between the interior and exterior. That means cell can respond to the environmental signals or can communicate with other cells through the molecular machineries present in the plasma membrane. Selectively, it is selectively permeable. Choose what may cross the membrane, just like a boundary wall. So there are some gatekeeping system, so selectively permeable. Fluid mosaic of lipids and protein. It is called fluid mosaic theory. That means cell membrane is composed of lipids and protein and like a mosaic in structure. And lipids are bilayer, contains embedded proteins. The plasma membrane is the boundary that separates the living cell from its non-living surroundings. This is simple, definition. The plasma membrane exhibits selective permeability, allowing some substances to cross it more easily than others. Phospholipids are the most abundant lipid in the plasma membrane. As I told you, plasma membrane is composed of lipid bilayers. So there are different kinds of lipids. Phospholipids are most abundant. Are amphipathic, containing both hydrophilic and hydrophobic regions. That means plasma phospholipids are amphipathic, containing both hydrophilic heads and hydrophobic region, that is tails. Head composed of phosphate group attached to one carbon of glycerol is hydrophilic. Two fatty acid tails are hydrophobic. So both hydrophobic and hydrophilic part, head is hydrophilic and tails are hydrophobic. We can see in the structure. Here you can see the clear illustration. Here you can see lipid bilayer. There is the head, hydrophilic, and interior, the tail, yellow color, hydrophobic. So the two bilayer we can clearly see. Singer and Nicholson in 1972 proposed that membrane proteins are dispersed and individually inserted into the phospholipid bilayer of plasma membrane. Here you can see very nicely phospholipid bilayer, and these are the just like mosaic. You can see some of the proteins are coming out from the lipid bilayer. So this protein, these are the receptors or the molecules involved in communication of the cells to the environment. So cell can crosstalk among each other in the multicellular organisms. So fluid mosaic model is already been defined. A membrane is a fluid structure with a mosaic of various proteins embedded in it when viewed from the top. Phospholipids can move laterally a small amount and can flex their tails. Membrane proteins also move side to side or laterally, making the membrane fluid. So here you can see freeze fracture studies of the plasma membrane support the fluid mosaic model of membrane structure. All are drawing, but by using different cell biological study. For example, freeze fracture study, scientists discovered the real structure of plasma membrane. Here you can see the proteins and one layer of lipid and another layer of lipid, and here is the cytoplasm. So a cell is frozen and fractured with a knife. The fractured plane often follows the hydrophobic inner of a membrane, splitting the phospholipid bilayer into two separated layers. The membrane proteins go wholly through the cells. Here is the fluidity of membranes. Here, a phospholipid in the plasma membrane can move within the bilayer two ways. Slightly they can move. Lateral movement, 10 to the power 7 times per second. So they can laterally move. And they can also flip once per month. Flop can move up and down, and here is the laterally they can move. So that means membrane structure is a bit flexible. The fluidity of membranes, if we can get more information, the type of hydrocarbon tails in phospholipid affects the fluidity of the plasma membrane. So hydrophobic tails, hydrocarbon tails contains different hydrocarbons. In case of lipid, unsaturated hydrocarbon tails with kinks, they can make the fluid structure of the membrane, and saturated hydrocarbon tails a bit viscous. So depending on the structure of the lipid, the fluidity of membrane is vastly regulated. The steroid cholesterol has different effects on membrane fluidity at different temperatures. Here you see in the membrane, cholesterol is a steroid. It can be, present in the lipid structure, and it helps the membrane fluidity at different temperature. That means higher temperature, it would be higher, fluidity would be higher. Membrane proteins and their functions. The membrane is a collagen of different proteins embedded in the fluid matrix of the lipid bilayer. Here you can see how complex structure is membrane. So there are some glycoproteins. These are glycoproteins. Carbohydrate also can be there, different. And glycolipid also. Extracellular side of membrane, there are some extracellular side of membrane are also present. Here you see cholesterol molecules are also present in the lipid bilayer. Microfilaments of cytoskeleton. Later I shall, this rope-like structure are cytoskeleton, and they are microfilaments. And microfilaments present just beneath the plasma membrane, hold the cell. So they are also very close to interior, close and interior part of the cell. Peripheral protein, and let me see. And there are many other protein molecules present in the membrane. So membrane structure is elaborately been studied and our understanding is very clear. And cell membrane is very crucial for even exchange of molecules, as well as respond to the environmental stimuli and defense of the cell from the stresses. And also, when you take a drug, there should be a receptor on the cell membrane that perceive the drug molecule, and you will get the action. So types of membrane proteins: integral proteins penetrate the hydrophobic core of the lipid bilayer. They are called integral proteins. Are often transmembrane proteins. There are some are transmembrane, you see, from outside of the cell and even inside the cell, cross the membrane. They are called transmembrane protein. N terminus is here and C terminus is here. And this type of membrane is like G protein couple receptor. They are function as a receptor of external signal. So this is they are very important. And in the eukaryotic system, G protein coupled receptors are very famous, and scientific discovery of G protein coupled receptor was awarded Nobel Prize, because nearly 60% of the drug molecules we are taking, they are working through the G protein coupled receptor. Peripheral proteins are appendages loosely bound to the surface of the membrane. Here you can see, not transmembrane, peripheral proteins. So 6 major functions of membrane proteins. What are those? One is transport. Transport, here you can see a molecule from inside to outside or outside to inside. So a protein that spans the membrane may provide a hydrophilic channel across the membrane. You see, channel is opening. That is selective for particular solute. It is selective, not any molecule can pass through. Other transport protein shuttles a substance from one side to other by changing the shape. Here you can see, first open and then close this side, and it is out. And this type of transport requires the energy from interior to exterior. And it is called active transport. And some transport is passive, no need energy. So enzymatic activity, one is transport, another is enzymatic activity of the membrane protein. A protein built into the membrane may be an enzyme with its active site exposed to the substance in adjacent solution. Adjacent solution, it can be inside here, it can be this transmembrane protein can act as an enzyme and catalyze the reaction. So membrane are organized as a team that carries out sequential steps of metabolic pathway. So they can participate in the metabolic pathway. Third, signal transduction. A membrane signal transduction, as I told you before, G protein coupled receptor or like that. A membrane protein may have a binding site, specific binding site with a specific shape that fits the shape of a chemical messenger. So, for example, we use the drug molecule. Drug molecule must have a specific must have a specific site that is called receptor. This type of protein is called receptor. And fits shape the chemical messenger such as hormone. And the external messenger may cause a conformational change in the protein. When it binds, there are conformational changes taken place and message goes to interior. And there is a defined signal transduction pathway. And this type of membranous protein is called receptor. receptor. To perceive the external signal and pass through the message to the interior, and very, very important. A cell-to-cell recognition system. Some glycoproteins present serves as identification tags that are specifically recognized by other cells. Then intercellular joining. You see, there is a two-cell neighboring cell in case of multicellular organisms. So intercellular joining, the protein involves also is called gap junction or tight junction. So junction, two cell holds together, especially in the tissue. Attachment to the cytoskeleton and extracellular matrix. Here see, some of the proteins are involved in attaching the cytoskeletal protein at the interior side, and the exterior side, it is attaching the extracellular matrix. These are the extracellular matrix polymeric substances. So in total, 6 types of proteins are involved in the membrane, and they do the defined function. The role of membrane carbohydrates in cell-to-cell recognition. So in the membrane, I told you there are some carbohydrates, that is glycosides or like that, and they help cell-to-cell recognition. Cell-to-cell recognition, one cell can recognize others. It is a cell's ability to distinguish one type of neighboring cells from another. Membrane carbohydrates interact with the surface molecules of other cells, facilitating cell-to-cell recognition. Synthesis and sideness of membranes. Membranes have distinct inside and outside faces. Already you have seen in the images. This affects the movement of proteins synthesized in the endoplasmic endomembrane system, Golgi, or endoplasmic reticulum. Synthesis and sideness of membranes. Membrane protein and lipids are made in endoplasmic reticulum and Golgi apparatus. You see, all the proteins which are involved in plasma membrane, they are synthesized in the endoplasmic reticulum and Golgi apparatus. So when a cell goes for division, before that they have to, the cell has to prepare the enough adequate amount of protein they need to synthesize in the endoplasmic reticulum and Golgi apparatus. In the cell unit, endoplasmic reticulum and Golgi apparatus, they produce the protein, and finally they transport from Golgi apparatus is called a packaging unit, and endoplasmic reticulum actually synthesize and then transport to the Golgi apparatus. Golgi apparatus package the proteins and materials in the vesicles. Vesicles is like a packet or like that, and moves to the cell membrane for plasma membrane for their use. Membrane permeability. Membrane structure results in selective permeability. A cell must exchange materials with its surroundings, a process controlled by the plasma membrane. And you know that hydrophobic molecules and polar molecules, especially are lipid soluble and can pass through the membrane rapidly, especially hydrophobic one, because membrane is composed of lipid, and hydrophobic molecules, those molecules dislike water, they can easily dissolve in lipid membrane and can pass through the membrane rapidly. Polar molecules do not cross the membrane rapidly. In that case, it requires, due to hydrophobicity, requires the channel. Transport proteins allow passage of hydrophilic substances across the membrane. Here you can see how they do. So one method, for example, this molecule is hydrophilic. In that case, there needs a channel to pass through from the cell. Uh, there are two types of transport. One is called passive transport, another is active transport. Passive transport is diffusion of a substance across the membrane with no energy investment. Carbon dioxide, water, and oxygen can easily diffuse across the plasma membranes. Diffusion of water is known as osmosis. You know osmosis very well. So simple diffusion here, in the osmosis experiment, you can know from the higher concentration to the lower concentration how it can pass through. So is the tendency for molecules of any substance to spread out evenly into the available space, move from high to low concentration down the concentration gradient, and this is the universal. So effect of osmosis on water balance. Yes, osmosis is the movement of water across a semi-permeable membrane is affected by the concentration gradient of dissolved substances called the solutions, tonicity. And tonicity is the ability of a solution to cause a cell to gain or lose water. And it has a great impact on cell without walls. Three states of tonicity: hypertonic, isotonic, and hypotonic are classified. So, for example, hypertonic, here you see high concentration of water, but water is, coming out from the cell. For as a result, cell becomes shrink. And here water is going in, and here hypotonic means water going in and dissolving the interior. So isotonic solution, possibly you know that if a solution is isotonic, the concentration of solutes is the same as it is inside the cell. So cell outside and cell inside, and the concentration of the solute is same. There will be no net movement of water. If it happens, then there will be no movement, no osmosis will be taken place. In that case, it is called isotonic. Hypertonic. Hypertonic means here you can see the concentration of the solute is greater than it is inside the cell. So the cell will lose water. In that case, cell will lose water, and losing water of a cell is called plasmolysis. And hypotonic solution, the concentration of solutes is less than it is inside the cell. In that case, you see the solvent will go, water will go inside, and the cell will gain water. It is called hypotonic environment. So water balance in cell without walls. For example, our blood cell has no wall. So animal cell. An animal cell fares best in isotonic environment unless it has special adaptation to offset the osmotic uptake or cross water. So water balance of cells with walls. Cell wall help maintain water balance in case of plant cell. So plant cell can maintain the water balance because it has strong cell wall. But in case of animal, especially human, if you stay a longer time in the water, you can see your skin will shrink, and due to the loss of water from the cell. Turgor pressure is the pressure of water inside a plant cell pushing outward against the cell membrane. Turgor pressure. If a plant cell is turgid, it is a hypotonic environment, and it is very firm and healthy state in most plants. If a plant cell is flaccid, it is an isotonic or hypertonic environment. So you see in case of plant, although cell wall protects or maintains water balance, even though if there is a drought, you can see the wilting and other problem. Water balance in cell with walls, especially plant cell, are turgid, firm, and generally healthiest in a hypotonic environment, where the uptake of water is eventually balanced by the elastic wall pushing back on the cell wall. Here you can see the turgid normal one, flaccid. Uh, 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 there are some water in and out, and in case of plasmolized, that means water is out only. So how will water move across semi-permeable membrane is a question. Solution A has 100 molecules of glucose per ml. Solution B has 100 molecules of fructose per ml. How will the water molecules move from solution A to B? If there is a question, there will be no net movement of water because the concentration of solute in each solution is equal. How will water move across semi-permeable membrane in case of solution A has 100 molecules of glucose per ml, solution B has 75 molecules of fructose per ml. So how will the water molecules move? In that case, there will be net movement of water from solution B to solution A, because solution B has, until both solutions have equal concentration of solute. So there will be a movement of the molecules. Solution A has 100 molecules of glucose per ml. Solution B has 100 molecules of sodium chloride per ml. Here you can see different types of molecule. In that case, each molecule of sodium chloride will dissociate to form sodium ion and a chloride ion, making the final concentration of solute 200 molecules per milliliter. So therefore, there will be a net movement of water from solution A to solution B, because here double number of molecules or ions are there. So movement will be there. So facilitated diffusion. There are facilitated diffusion is a type of passive transport aided by proteins. So diffusion can be aided by the protein. So protein can function to facilitate the diffusion. In facilitated diffusion, transport proteins speed the movement of molecules across the plasma membrane. So there are some other proteins I told you, channel protein. Here you can see, as I told you, this is hydrophobic. Even polar molecule from extracellular fluid can enter into the cell through a channel. So if, for example, sodium channel or calcium channel or like that, so the channel has specific passage. So any ion cannot pass through the specific channel. So specific channel for passing through specific ions. And there are some carrier proteins also. Here you can see carrier proteins, how carrier proteins also can facilitate exchanging of molecules from exterior to the interior, very nicely shown here. So active transport uses energy to move solutes against their concentration gradients, requires energy usually in the form of ATP. For example, the sodium potassium pump, there are a pump protein. I told you how protein helps to influx and efflux of molecules. The influx means molecule in and efflux means molecule out. In case of calcium also, there are some transport proteins are involved, and they help to make a balance, homeostasis in the ionic homeostasis in the cells, and it requires the ATP. So here you can see the passive transport directly can do, but in case of active transport, it requires the ATP. Maintenance of membrane potential by ion pump. Yes, ion pump membrane potential, because when concentration in the interior is increased significantly, and the exchange of influx and efflux of molecules are also involved in the signal transduction pathway. So membrane potential, electrochemical gradient, and electrogenic pump, they are also involved in the membrane potential by ion pumps. Proton pump, there are also specific protein involved in exchange of protein, and they are also active system. For example, sometimes acidity increased in the cell significantly. In that case, proton is pump out from interior to the exterior of the cell through the proton pump. That means proton pump is also a protein. Co-transport sometimes occurs when active transport of a specific solute indirectly drives the active transport of another solute. Involves transport by a membrane protein and driven by a concentration gradient, obviously. So example of co-transport, here you can see protein. Proton is transported here, and in that case, sucrose molecule can also pass through co-transporter. Co-transporter also require, are also they are also protein, so they are also involved in the exchange of the molecules. Bulk transport across the plasma membrane occurs by exocytosis or endocytosis. Large proteins are involved there, cross the membrane by different mechanisms. In exocytosis, transport vesicles migrate to the plasma membrane and fuse with it and release their contents. That means a vesicle. Vesicle is like a putli, something wrapped up, maybe protein or other things. So can be also passed through the exocytosis for making the cell wall or like this. In endocytosis, the cell takes in macromolecule by forming new vesicles from the plasma membrane. So these are the endocytosis. Here you can see how it happens. Yes, phagocytosis and moves to the lysosome, and then it dissolved. Lysosome is the dumping place, and it is dissolved like this way. So endocytosis can be taken place. Exocytosis also can be taken place. Here you can see how vesicles are going out from the cell. So 3 types of endocytosis have been recognized. Phagocytosis, a cell engulf a particle by wrapping pseudopodia, or like this, the pseudopodia. These are the electron micrographs, and these are the drawing. Or pinocytosis, droplets of extracellular fluids can be, engulfed and pass through the interior as a vesicle. So these are the process. So this is all about the cell membrane and their functions and their structures. Do you have any question?
I learned a lot. Today I learned a lot of things. It's a really interesting topics, and also simple things, but it's a lot of information.
Thank you very much. And I tried to give you the easiest and way to understand the cell membrane and their function. Actually, they are more complicated, but the basic and fundamentals of cell membrane, their structure and function, if you go through these slides, it would be enough. And for further information, you can go through the book Cell and Molecular Biology by Alberts and co-workers. That book is a fantastic one. And if you have any question, then you may ask even me later on. And please go through this slide. And I am, you received this slide, right, before?
Yes, sir.
So please go through these slides. And tomorrow I shall also tell you in detail about the cell cytoskeleton, because you know the plasma membrane now and cell cytoskeleton, how as cell structure it is maintained, and they hold the they are basically proteins, and there are three types of proteins involved in cell cytoskeleton, and you can understand how mysterious a cell is, how cell biology is. Basically, in our agriculture, the curriculum of agriculture, cell biology is ignored, which is very essential for understanding the biology of living organism. Thank you very much. Then please enjoy the lectures, and I shall also give you the recording so that you can repeat my lecture and go through it repeatedly. All the best wishes.